In this video, we're going to cover some basic eye anatomy to help you become familiar with the structure of the eye so that you can start to understand how these structures are used to allow for vision to actually occur. If you look at this picture that I have here, what we have is an eye that has been cut sagittally so that you can see the internal structures of it. So I'm going to go through some of these structures, talk about what they're used for, to help you start to come to understand how the eye actually works as a functioning organ to allow vision to occur. The first thing that you should know about the eye is it has a thick and sturdy wall, and that thick and sturdy wall there is to protect, of course, the eye, but it also maintains the shape of the eye. There are 3 layers to this wall. So if you look at where I'm running my pointer right now, this outermost layer is what's known as the sclera. And on the back part of the eye and kind of the more lateral parts of the eye, this is composed of a white, thick, strong connective tissue. So when you look at somebody and you see the whites of their eye, what you're actually seeing is the sclera. Right here, this portion of the sclera that sits in front of the colored region of the eye, so the central portion of the sclera right over the iris, is what's known as the cornea. In this particular area, the structure is a little bit different. So the cornea is actually clear, it's translucent, and the reason for that is light rays have to be able to pass through it. So in that particular area of the cornea, the outermost layer of the eye wall is a little bit different. It's translucent. If you look at the rest of this picture, we'll go in a layer. So we've talked about the outermost layer, the sclera. Here we've got a layer that's represented in red. This is the middle layer of the eye. It's what's known as the choroid. It has a really high density of blood vessels. And just inside of that, we've got the innermost layer of the eye that you can only see here in this posterior part of the eye. And that's the neural layer, or what's also sometimes referred to as the retina. So those are the 3 layers of the eye that make up the eye wall. If you look at the eye as a whole, you'll notice that it's divided into 2 different chambers. So we've got an anterior chamber here on the front side, just behind the cornea, and it is filled with a fluid that's known as aqueous humor. And aqueous humor is just kind of a liquid, almost like water. It's being produced all the time and released into this anterior chamber of the eye. And it's also draining out at all times through a little structure that sits right here, which is known as the scleral venous sinus. The role of this aqueous humor is it's going to help to pressurize the front of the eye, because it's very important that the front of the eye, and particularly the cornea, maintains its shape, because the shape of the front of the eye is going to allow it to properly bend light rays as they move through the eye, as you're going to learn about in the activity that we do this week. The posterior part of the eye is also a chamber, so it's known as the posterior segment. You can see that back in here. And this posterior segment is also filled with a fluid. But the fluid that's in the posterior segment is more gelatinous. It's thicker than the aqueous humor, and it's known as the vitreous humor. The vitreous humor is really important for pressurizing the back of the eye and helping it to maintain its shape, which is very important to being able to see clearly. But the other thing that the vitreous humor does back here that's really important to the function of the eye is it keeps this innermost layer that we talked about, the retina, actually pushed up against the back of the eye. There is nothing that physically attaches the retina to the other layers of the eye wall. It's just the pressure from this vitreous humor that kind of holds it in place. And that's really, really important, because it's within the retina that we have all of the photoreceptors, all of the neurons that respond to light and allow for vision to be able to occur. As light is entering the eye, it comes first through the cornea and then through the anterior segment. It travels across the lens, which you can see here, and then back through this vitreous humor, and it lands on the photoreceptors that are located within the retina. Without that vitreous humor there, the retina can actually fall, and this is an injury that happens in people sometimes. And when it does, they lose part of their vision because they've lost the photoreceptors that would otherwise be picking up the light that's traveling through the eye and landing on the retina. So we've talked about the wall of the eye. We've talked about the two different hollow chambers that are filled with fluid in the eye. What I want to do now is kind of focus on these structures in here that sit really in between the anterior segment and the posterior segment. So the first of these that you can see right here, this is really an outcropping of that choroid, the middlemost portion of the eye. This is what's known as the iris. So it's the colored portion that you see when you look at somebody's eyes, whether they have blue eyes or brown eyes or green eyes. That's the iris that you're seeing. Right in the center of the iris, there is a little black dot, and that's the pupil. And the importance of the pupil is it's going to let light rays pass from that anterior segment into our posterior segment and through the lens of the eye. So just behind the iris and the pupil, we've got the lens, which you can see right here. This particular structure works along with the cornea to bend light rays and focus them so that they land directly on the retina. The lens is, at least in a healthy young eye, very flexible, and it can change its shape. And in fact, it needs to be able to change its shape as you go from distance vision to close-up vision. So the lens is changing shape depending on how close objects are that you're looking at. And because it needs to be able to do that, it has a muscle. So you can see that muscle here. It actually surrounds the lens 360 degrees. So you can see it back there as well. Up in here, we've cut the eye sagittally, or it would cover the lens here. So it's 360 degrees around that lens. And that particular muscle that helps to bend the lens, to change the shape of the lens to accommodate for vision, is what's known as the ciliary body. The other anatomical structure that you should be familiar with as we're looking at and just first getting familiar with the structure of the eye are the suspensory ligaments. So you can see these in here. You can see them down here as well. They would be actually 360 degrees all the way around the lens. They are what attaches the lens to the muscle, the ciliary body, so that that muscle can contract or relax to pull on or release pressure from the lens in order to change the shape of the lens and again accommodate for vision. What I want to do now that we've talked about some of the major structures associated with the eye is I want to focus in much more on the retina. So that's that neural layer, again, where we've actually got the photoreceptors that are picking up light and responding to light and allowing for vision to occur. So if you look at this picture here, again, we've got a sagittal section of an eye similar to what we saw on the last slide. The artist has taken a segment of the back of the eye that includes the neural layer, the retina, and they've magnified it much more highly. So you can see that here. If we were to take even a smaller section of the back of the eye and put it under a microscope and actually look at the cells that make up the sensory layer here in the back of the eye, what we would see is a structure that looks somewhat like this. So this is an artist's representation of the sensory layer of the eye, or the retina, under a microscope. And I want to talk about some of the cells that are found here in the retina and what those cells are doing to help allow for vision. We're going to start in the most posterior, so this would be the backmost portion of the eye that we're looking at. And here we have a single cell layer, which is known as the pigmented layer. So you can see it represented here, kind of in this tannish color. The role of the pigmented layer is it's going to help to nourish the photoreceptors and kind of protect the photoreceptors. So if we go just anterior to the pigmented layer, we have a layer of cells right through here that's represented in purple and yellow. These are the actual photoreceptors, so the cells that are picking up light rays and generating action potentials in response to them to allow vision to actually occur. So you'll notice some of these cells are represented in purple, and they have kind of a rod shaped into them. And other cells that are interspersed in this photoreceptive layer are represented in yellow, and they have more of a tapered tip or a cone-shaped tip to them. These purple cells with the rod-shaped end are known as rods. So that's where they're actually getting their name from, is just their structure, what they physically look like. Same thing with the cones. So these yellow cells that we're seeing that have this tapered end are referred to as cones. And together, our rods and our cones are our photoreceptors. There are some differences between these two cell types that you should be aware of. So first of all, they both respond to light, and they send action potentials to the brain in response to light, and that allows us to be able to see. However, rods are much more responsive to light than our cones. Rods are able to be activated at very low levels of light. So when you are stumbling around your house at night with maybe just a nightlight, or you're out walking at night through just moonlight, or you're in some other dark type of situation, light levels are so low in those situations that cones cannot respond, but the rods can, because they're so much more responsive to light. One of the things that's kind of unique about rods, though, is they do not allow for color vision. So if you've never noticed this before, you will now. If you are stumbling around your house, or you're driving in your car late at night, under moonlight conditions, something like that, you see grayscale. You're able to see really just black and white, gray, those kinds of things. And the other thing about the rods is they do not give you as sharp as images as the cones do. So things tend to be more fuzzy when you're working and living in low light conditions. The cones, again, are these yellow ones with the tapered ends. They are not as responsive to light as rods are, so it takes much higher levels of light to be able to activate cones. So think broad daylight conditions, or working with a lamp on that's bright enough to give you clear, colored pictures. That's what cones are capable of. So they're what allows for color vision. They also give you sharp images that aren't so fuzzy. You can see as you look at these photoreceptors that the rods are more common in the eye than the cones actually are, and that is the case. However, when light travels through the eye, so you can see this little diagram here that's indicating the pathway of light, it goes all the way to the back. I'm going to jump back up to this microscopic picture of cells in the back of the eye. The light has to pass through 3 layers of cells in the retina and come here to our photoreceptive rods and cones to cause those photoreceptors, the rods and the cones, to generate action potentials in response to that light. When rods and cones generate these action potentials, that's going to send a signal to this next neuron in the pathway, which is known as a bipolar cell, which sends a signal to this most anterior cell in the pathway, which is known as a ganglion cell. And these ganglion cells feed into what's known as the optic nerve, so the nerve that's taking away all this information on vision from the back of the eye. So one of the things that's kind of weird is that light travels through the eye in this direction. It's going to activate almost our most posterior layer of cells, and then electrical signals move away from those cells in this direction, opposite of the pathway of light, as they're feeding into the optic nerve, which exits the back of the eye. We've talked quite a bit about the basic anatomy. We've spent some time looking at the microscopic neural anatomy and the cells that are actually located there to allow for vision to occur. There are a couple of other structures that are associated with the sensory layer, the neural layer, the retina, whatever you want to call it, that are worth mentioning, because you're going to be learning about them further in the activities that you're doing for this particular folder. So the first of these structures that I want you to be aware of is what's known as the optic disc. If you look at this picture here, the optic disc is this area right here, which is directly in front of where the optic nerve exits the back of the eye. This particular area has no photoreceptors, so there are no rods, there are no cones in this particular area. We go up here, we're looking at an actual picture taken of the back of somebody's eye. So we're seeing the retina. Here is where the optic disc would be. So this is where the optic nerve exits. It's also where blood vessels are entering and exiting into the back of the eye. And again, in this particular area, there are no rods and cones. This causes a situation which is known as the blind spot. So all of us out in our peripheral vision on both sides, so on our left eye and our right eye, we have what's known as a blind spot. This is something that we're not typically aware of, so you don't normally notice that it's there, that you have this area where you're blind, where you can't see anything that's out in your peripheral vision. And the reason for that is the brain is very good at kind of filling in the missing information with something. So it makes you think that you're seeing something, and it kind of fills it in based around the patterns of what's out in your peripheral vision, even though there's no light rays and there's a blind spot out in your peripheral vision. You're going to have a chance in the activity that you do in this particular folder to check your blind spot and find where it is. But it exists because light rays that are coming from objects which are within your blind spot are light rays that are landing on the optic disc of the eye, where there are no photoreceptors to actually be able to pick them up. If we go just lateral to the optic disc, so again, where that nerve is exiting the back of the eye, you'll notice we have a small kind of darker colored area of the retina, which is known as the macula lutea. And right in the center of the macula, there is a little small depression. So it's only about 2 millimeters across, and that depression is known as the fovea centralis. The reason that this area is important and worth mentioning is because it is the area of the eye where you have the highest density of cones. So it's estimated that about 50% of the cones in the retina are located specifically within the macula. The macula, and specifically the fovea centralis, are responsible for what's known as your central vision. So central vision is what you can see directly in front of you that's sharp, that's in focus. If you take a second and look at something out in the distance, you'll notice that whatever you're directly focused on is very clear, but just outside of that, most of the things that are in the peripheral vision are really fuzzy. And the reason for that is when you look at an object and you're focused on an object, the light rays that are coming from that object, when you focus on it, are coming into the fovea, where you have a very high density of cones. Light rays from all of the other areas of your peripheral vision are landing on some other area of the retina where there are not as many cones to pick them up, and because there are not as many cones, the images aren't as sharp, and that gives you kind of a fuzzy peripheral vision just due to the lack of cones in other areas of the eye.